episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor. Welcome, everybody, once again to another episode of our show with another really fascinating guest for you today, helping to create a better tomorrow on many different fronts. Today we have the opportunity of being joined by Dr. Spring Behrouz, who is the Chief Executive Officer of a company called Vincere Biosciences, which is a biotech company focused on developing novel small molecule therapeutics, ultimately targeting mitochondrial pathways and the improvement of mitochondrial quality in certain cells. She is also the CEO of NeuroInitiative, which is a computational biology company that she also co-founded, which develops simulations of various diseases using their patented software platform. And the focus of our research there has been on the elucidation of complex converging pathways that contribute to the pathogenesis of Parkinson's disease, this horrible neurodegenerative disorder we talked a little bit about in past episodes, affecting 1 million Americans, over 10 million people around the world nowadays. Dr. Behrouz received her graduate training Michigan State University in the lab of Dr. John Goudreau. We're studying differential susceptibility of dopaminergic neuron subtypes and models of Parkinson's disease. She completed her postdoc training in the laboratory of Dr. Matthew Frazier at the Mayo Clinic in Jacksonville, where she was focusing on a variety of in-vivo and primary cell culture models of Parkinson's pathogenesis. She was part of the team that discovered the pathogenic mutation VPS35. A lot of really exciting things to talk about today. Dr. Spring Behrouz, thank you so much for taking time out of your schedule to talk to us.
Good morning, Ira. Thank you for having me.
It's great to have you, and as we typically do, I'd love to start off by handing you the floor just for a few minutes to further introduce yourself a little bit. If you could sort of take us back a little bit about everything from where you grew up, when you first got interested in science and neuroscience, and a little bit of your early path on the way to what you're doing now. I think that'd be a great way to start off.
Yeah, sure. And thank you for the very thorough kind introduction. I think for me, one thing that has been very constant from the time I was very small to now is wanting to work on big problems that had large impact on humanity, really things that can actually alter the course of how humans live in the future. And there aren't too many of those. There are only a handful, or maybe 10 I could count, that are really large enough to be impactful at that level. And initially I thought I might go into physics, but I ended up going into biology. And within our field, I think, aging and diseases of aging are the ones that can have the biggest impact. Obviously, the aging itself is something that we all deal with, and it's something that for a long time, I even myself thought was kind of mandatory and not modifiable. And finding out that it is modifiable is really exciting. And it's now starting to really gain traction, and we're starting to try to translate that to humans. We've had a lot of success in animal models. And then diseases of aging like Parkinson's disease, of course, is a primary thing we work on. And where I started with Parkinson's disease. And when we formed the computational company, the idea was all of this new data that's coming out now, more than gigabytes, terabytes of data just becoming available on a regular basis. And it's just continuing to increase as with all this omics data that's becoming available, transcriptomics, metabolomics, whatever, proteomics, and trying to make sense of all of that data. You really need the computational advances that have been made that are making huge advances in other areas. If you look at, for example, facial recognition software, how good is that? It's just fantastic. So being able to translate that into biology and drug discovery in particular has been one of the main goals. And that's really what honed us into, you mentioned that we work on mitochondria. That's really what honed us into mitochondria as an area of importance for Parkinson's disease. And we've started to develop small molecules that we can actually take to the clinic and take to the patients. So starting all the way from, the very, very kind of big picture theoretical to now having small molecules that hopefully we can take to the clinic very soon.
Very exciting. And we've touched on mitochondria a bit on the show in the past, and obviously everyone listening to the show that's had sort of, biology 101, a little bit about the power plant of the cell story. But when we think about mitochondria, we don't usually think that mitochondria has different degrees of a task to do depending on what cell we find them in. Some cells have very few. Some cells, like what you work on in terms of the neurons, have a tremendous amount of them. Unlike other cells, the neurons hang around for a long time. So there's this really interesting, we don't think about it, but this interesting imbalance between the cell and how much protein is turning over a lifetime. Talk just before we get into sort of mitophagy and some of the things you're targeting. Talk a little bit about how you think about mitochondria in general and the neuron, because it's a little different than just, hey, it's just providing some energy. Doing a lot more here.
Yeah, absolutely. I do love that, there was a meme going around with all the kids about mitochondria as the powerhouse of the cell. And it was, it's kind of a joke, but I love that all the kids know that mitochondria at least are the powerhouse of the cell.
Which I know.
But beyond that, the way that you're taught is sort of this oval shaped thing, and it's very static. And you have, usually there's one pictured in the cell and it provides the cell with energy, and that's the extent of it. But they are really dynamic. The mitochondria are so incredibly dynamic. And if you look at, videos, I highly encourage people to just, Google or look up on YouTube some videos of mitochondria. So incredibly dynamic, and they move, within your cells. They function they kind of like their own little independent organisms, which they were many years back, right? They sort of co-evolved with us. And if you think about them in that way, and they can just like bacteria, they can split up. So we call it mitochondrial fission. They can fuse together, mitochondrial fusion. They move around the cell, so they get transported. They can attach to other organelles so that they can provide the energy very much directly next to those other organelles. So for example, lysosomes, which break down things inside your cell, so they can attach right to those. And then they don't live forever, even just naturally. They are inside your cells, particularly brain cells, around 30-40 days. So they need to be broken down and cleared away, and the materials are making new mitochondria, and the cell helps to make those new mitochondria. So that process is incredibly dynamic, and being able to manipulate that process has a lot of potential. And we're working, as you mentioned, on mitophagy, which is clearing of these damaged mitochondria, and kind of the repair machinery, because you can take even like small chunks of the mitochondria and recycle those rather than taking the whole mitochondria, because that's very energetically expensive for the cell. But all of the different pieces can be modified. And I think it's, it provides us with a lot of potential, because mitochondria are incredibly important, not just for diseases of aging like Parkinson's disease, but for the process of aging itself. So for example, if you overexpress some of these mitophagy proteins, PINK1 or Parkin as examples, you can extend lifespan and health span in fruit flies by about 30%. So they live longer and they're fertile for a longer period of time, suggesting that they're actually healthier. So this is, it's really exciting stuff to be working on.
And yeah, it's you mentioned mitophagy, and we've talked about autophagy in the past, or the selective degradation of the cell itself. Here, it's a little more specific. The whole theme is about, okay, if we can get rid of they've done their job. Some are, don't have as high quality as others. We can get them out of the way and restore this homeostasis, which is obviously a very important process in keeping us balanced on all fronts. Talk a little bit about what you mentioned Parkin, and then also like looking through some of your papers and materials, something called USP30, where there's some enzymes that are both involved in sort of speeding up and slowing down this repair and recycling mechanism. Talk a little bit about sort of this mitophagy ecosystem that you study, and some of the different targets that you look at in either enhancing this process or stopping if it's going too far.
Yeah, absolutely. So the trigger of how the cell knows which mitochondria are damaged to actually clear them, because the cell doesn't want to clear healthy mitochondria that are functioning properly. So there has to be some kind of a signal to the cell, right? That lets you know, okay, this is damaged mitochondria, clear this one. And the mechanism, as you mentioned, is through autophagy, although this is a selective form of autophagy. So the mechanism is that there's a protein called parkin, which is an E3 ligase. It is recruited to the cell via another protein called PINK1. And this PINK1 protein normally would get just pulled through the mitochondria and cleaved if the mitochondria are functioning properly. But if the mitochondria are not functioning properly, it stays on the outer membrane of mitochondria and it starts to accumulate. Then it recruits parkin, and parkin starts to add what are called ubiquitin chains. So just a whole bunch of ubiquitin tags right onto the mitochondria. And that's really the signal for the autophagic machinery to come and clean this up.
Got it.
So once you have that, then the autophagic machinery understands that this is a mitochondria that needs to be cleared away. And the same actual mechanism is used for repair systems too. So let's just say you have a small portion of the mitochondria that needs to be taken away. So just very localized damage that can be taken away. It's the same machinery that's doing that. On the other side of it, there's always a yin and yang in biology. You know, what goes up must come down. So on the other side of parkin that's adding these ubiquitin tags, there's a protein called USB30, which is right on the mitochondrial membrane, always on the mitochondrial membrane, doesn't need to be recruited. And it takes these ubiquitin tags off. So it sort of works against parkin. And they're the yin and yang of each other. So, you know, parkin puts them on and USB30 takes them off. And so you can manipulate the system by enhancing the activity of parkin, for example, and trying to get these ubiquitin chains on more quickly, enhancing mitophagy. Or you can lower the activity of USB30 and thereby accomplish the same thing. If you think about it as, you know, parkin is the gas, so you can press on the gas pedal, or USB30 is the brakes, take your foot off the brakes, right? Or you can do a combination of the two.
Excellent.
And there's a video, there's a video on our website too that explains this in more of a, you know, pictorial format.
Yep. We'll definitely link to all that in the show description. But, you know, I'm thinking through, you know, you talk about parkin, USB30. In the beginning, I talked about some of your past work on some of these genes like LRRK2 and VPS35. But this is all a very small component of, you know, all the genes and all the possibilities. You mentioned transcriptomics and proteomics, everything else that's going on inside here. Alongside of what you're doing with mitophagy, you know, you've developed this very powerful other engine, this NeuroInitiative, where you're using some of these advanced tools that we talked about a bit on the show, things like in silico study and artificial intelligence. Obviously, the CNS probably the most complex of all our systems, so I don't want a different guest to fight, but talk a little bit about the NeuroInitiative and how some of these new tools and also maybe some of the future tools that you're thinking about assist in really dissecting this complexity of the CNS.
Yeah, and you know, we named it NeuroInitiative because obviously we're interested in neuro, but the technology can be used for any modeling any kind of cellular system and any kind of disease. So we probably changed the name at some point. And I can talk about the computation probably for the entirety of this time. So just let me know which areas are interesting. But the one of the things that we developed early on is a simulation system, which is kind of different from traditional AI in that it creates a virtual three-dimensional space. So if you think about, you know, a 3D space virtually, and then you can put in a cell, and then you can put in organelles inside that cell. For example, you can put in mitochondria, lysosomes, whatever. And then you put in the proteins, lipids, small molecules, all of the things that should normally be in a cell, you put in there. And each of those is associated with rules, biology rules. When they come in contact with each other, they can act according to those biological rules. And everything moves around according to laws of physics, which that part was actually much easier because a lot of people have done physics type simulations, right? So you make this kind of static thing and then you allow it to play over time. So you're adding the fourth dimension of time in there, and everything moves according to Newtonian laws of physics, and then they interact according to rules of biology when they come in contact with each other. So because of this, you can have fission, fusion, things moving around, being transported, neurotransmitter being released, all of that. And it's really, really interesting and exciting. And then what you can do is you can take transcriptomic data, let's say from a Parkinson's disease patient and from a healthy control, and you can compare those and see what's different about them, right? And you can do this, you have the level of granularity that you have. You basically see all of the proteins at every single moment in time. And we don't have the ability to do that in the laboratory. You can select certain time points and get the entire transcriptome, for example, but it's quite expensive. So you have to select very small, few time points to be able to do that. Or you can tag multiple proteins with GFP or different colors, so you can see them and you can actually visualize them moving, but then you're limited to how many you can actually tag. So in that way, we're able to see everything, and then we don't have to say, hey, is A different? Is B different? Is C different? It just tells you these are the ones that are different. These are not the ones. These are the ones that are the same. So it allows you to differentiate between, for example, a diseased cell and a non-diseased cell. And then more importantly, you can go in and say, hey, what if I add this manipulation? So what if I add a drug that can inhibit USB30, for example, which is what we're doing, right? What if we add that drug in? Is that likely to fix the problems in the system? And you can do that before you even develop the drug, because this is all virtual, right? Yep. So before we even go into the problem of developing a small molecule, developing a drug, we do this to make sure that what we're working on is actually relevant to the human disease. And even more exciting is that we have this transcriptomic data from brain cells of patients, yep. right? And we don't have access to that otherwise. We can get fibroblasts of skin cells. We can differentiate those into neuronal cells, iPSCs, even IDAs, which are dopaminergic neurons. But they're not. You tend to you reset them when you differentiate them, or sure. When you reprogram them. So really having access to the omics data and being able to play that out from brain cells of patients that are 40, 50, 60 years old is a really powerful thing that we don't have access to otherwise. So that's one aspect of what we do computationally, and that's a really unique thing to us. We patented it, and not many other people are working in this area, partially because it's rather difficult to do and is very resource intensive as well. On the other side, we're doing a lot of AI work as well, and those can be used for other things. I'm happy to talk about that more if that side is interesting.
Yeah, no, it's a fascinating, I'll say, convergent, skill set that you have there that is capable of looking at, as you were saying, it seems like it goes quite far beyond the CNS and the potential of the system. I was just interested, obviously Parkinson's is a sweet spot for you, and obviously some of the other major CNS disorders. Any interesting, obviously when you're in, nothing confidential, but when you're in the NeuroInitiative system, any interesting targets that pop up for other sort of orphan or rare CNS? Because I've been running in, as we talk about Parkinson's and Alzheimer's, I'm always running into researchers that have really interesting, I've never heard of these conditions, like a lot of the tauopathies is an example. I'm doing a show next week and I was coming up to speed almost. I didn't realize there were so many tau disorders, but any interesting things that you can talk about that outside of the big ones that you're finding in the NeuroInitiative system that, hey, that'd be interesting to go into, or maybe there's an interesting spin-off company here that we can do something with.
Yeah, absolutely. So, once we honed in on mitochondria and we're talking about clearing damaged mitochondria, yep. then you sort of have to step back and say, well, what else is that useful for? Because obviously mitochondria are essential parts of your system. Yep. And having the ability to clear damaged ones may be helpful, not just for Parkinson's disease, not just for aging, but for other specific disorders. And as you mentioned, there are rare diseases that are specifically due to mitochondrial problems. So, yep. Mitochondrial myopathies, malice, things like that. And yeah, absolutely, we look at those, but it's more in the context of kind of staying within the same pathways that we are already finding important. And, sure. We call it kind of a pipeline and a pill, basically saying, well, what else might this be useful for? And yep. some of those rare diseases are definitely areas where it might be useful. And in some ways, you may get a more homogeneous population of patients with those because they're more similar. Whereas Parkinson's disease, they tend to be relatively, it's a relatively heterogeneous. Each patient doesn't manifest the same way. They don't have the same genetic mutations. The cause for the majority of cases are, caused by things we don't know. They're called idiopathic. Sure. You have rare genetic forms of the disease, but these rare rare genetic diseases kind of give us a more homogeneous population of patients that we can test on. And also it is, from an FDA perspective, also sometimes it's easier to test on a smaller population of these patients, especially if they progress more quickly than, say, Parkinson's disease is a terrible disease, but it's a very slowly progressing disease. So you really have to follow the patients for a couple of years to know if your drug is making a big impact or not. Whereas with some of these rare diseases, you can maybe do that more quickly.
Outstanding. I noticed Vincere has, been very active on the partnering side. You have recently a $1 million grant from Michael J. Fox Foundation, SBIR at the NIH. You're in the J&J incubator in Cambridge, really building a nice sort of ecosystem around you. Any sort of next steps for the company that you can talk about? And are you raising private equity now? Any other interesting things that are going to be coming up in the next 6 months? Conferences you're going to be talking at, new TED Talks that we should be aware of?
So we are privately funded as well as grant funded. So if there are people who are interested in that, they should just contact us. We do talk to interested parties regularly. From a pharma kind of partnering perspective, we do work closely with a lot of the pharmas. It's nice to be in Cambridge, within a 1 mile radius. We really have everything that we need. We have Harvard and MIT right here. We have all of the VCs have offices here. Sure. All of the pharmas have offices here. It's been a bit of a strange year, the past year, as you can imagine, due to the COVID situation, but things are actually starting to be back to normal. And so we stay in touch with, a lot of the pharma companies because they're very skilled at taking drugs to the clinic, right? Sure. So we try to learn as much as we can from them. And our hope is to partner with one of the pharma companies to help us take it into the larger human clinical trials, because the phase 2 clinical trial for Parkinson's disease is very, very expensive and requires a lot of skill sets that we may not have in-house. We're much on the kind of invention earlier part of the process. So the goal is to partner with one of the companies once we are a bit further along. We kind of still like to get it a bit further along before we hand it off.
Absolutely.
As far as speaking engagements, it's been now I've been flooded with a lot of requests again, but it was kind of a lull for a little bit of time. But I'm sure there'll be a bunch of those coming up soon.
Yeah, especially with the progress that you're making and your portfolio, I can see.
In some ways, it's been nice to actually just be heads down and working and not going out. But I very much enjoy the conversations and conferences. So looking forward to it again.
Along your path in this particular space, you've no doubt met quite a few important people in industry and academia and government. Take a little time to mention, shout out to whomever you want that's been really instrumental on your path in developing Vincere, the NeuroInitiative, and all your other research programs.
Yeah, absolutely. I mean, first and foremost, our team at both Vincere and NeuroInitiative, my co-founder Andy Lee, and the rest of the team, everybody is fantastic. We have brought together skills from both the computational world and the drug discovery world, including medicinal chemistry, pharmacology, all the way to, on the other side of it, programming and AI and data science. So it's been really fantastic to work with these stellar group of people, and we always try to recruit only the top of the field. So it's been really fantastic. On the collaborator side as well, we've been very fortunate to have some good collaborations. And the funding, particularly from, I think, Michael J. Fox Foundation, has been fantastic because they not only give you funding, but they also give you tools and resources. They have consortia that allow you to communicate with other members of the team. And the Parkinson's community is very collaborative. And, we're kind of like a big family, which has been really nice because you can bounce ideas off of one another and not be overly competitive. So and then of course NIH for funding as well. They tend to be a bit more regimented, but definitely it's always appreciated. These kinds of non-dilutive funding is always appreciated.
Absolutely. Wonderful. So, I was just, wondering, on your Twitter page, in the sort of list of the stuff that you are, mentions neuroscientist, inventor, entrepreneur, CEO, got a lot going on. But the first thing you list yourself as is a nature lover, and I was just interested in what nature that was. But is that cellular nature, or are there other areas of the natural world that, when you're not thinking about all this science and Parkinson's stuff, that you enjoy? The ocean, the rainforest? What nature do you love?
I love all of nature. I mean, I think that we tend to kind of isolate ourselves. I often say I have two lives, one in the here, work life, and then the other life is just when you go out in nature and you're, alone with the trees and the birds and the squirrels. And I do tend to make friends with unlikely animals, which at some point is bound to get me killed, I think. I was recently scratched by a squirrel, which, was I tend to feed random things, and then they and try to pet them, which is not wise. I do not recommend doing that. But no, I mean, I think it's, as a biologist, I think it's hard not to appreciate nature, right? This massive system that if you just look at the planet Earth, and we think of Earth as, most people think of it as a non-living thing, but if you really think about it, we are the organisms within it that are communicating with one another, and we give rise to this much larger being, if you will. If you think about, from a neuroscientist perspective, each individual brain cell doesn't really have consciousness in the way that we think about it, right? Each individual brain cell doesn't, it only knows what it knows within the context of itself and its neighbors and whoever it's communicating with, right? But when you have millions, billions of brain cells that are communicating with one another, you get this consciousness of a human being or animal even, right? And so 1 + 1 does not equal 2, it equals something much, much greater. And that's what I get from nature, is each of these components that are working seemingly kind of separate from one another give rise to this much, much grander, more beautiful thing that we call planet Earth. But I think each individual thing is really fascinating. And then on the flip side, if you zoom way into it, and if you look at one cell, you see that within that you have the same thing. It's kind of a fractal thing, right? You have organelles that interact with one another, proteins that interact with one another, and give rise to this kind of self-sustaining cell. So really, it's just fascinating, and I think we learn a lot from nature. Biomimicry is an entire field that I think is incredibly fascinating, and we try to invent as many things as we can, but really the planet's been inventing things for billions of years, so we can just look and learn maybe something.
Yeah, it's a very elegant perspective, and I very much appreciate myself, the natural world, and I think sometimes we separate ourselves in this era of high tech a little too much from it. When I saw that in your bio, it's like I can appreciate that, and I like that that you can stay converged, just keep it all as one, and not get too reductionist with the thinking. That's exciting.
Yeah, absolutely. And just I think even if you don't think about it in a deeper level, like I just mentioned, if you're just in nature, there's a kind of a peace and calmness that's associated with it. I think everybody feels that. It's hard to deny it. So.
And it's especially useful for CEOs and entrepreneurs, especially in 2021.
Absolutely.
But, yeah. This has been a really fascinating discussion, and really I think you're on a wonderful path here. You definitely have all the tools, and putting the pieces together for this area of extreme unmet medical needs. So really wishing you the best with all of this, moving it forward. for everybody that's going to be listening to this particular episode on the podcast or watching on the YouTube channel, you've been listening to Dr. Spring Behrouz, CEO, Vincere Biosciences, CEO of the NeuroInitiative, doing really amazing things, Parkinson's disease, CNS disorders in general, mitochondrial disorders. Definitely check out her companies, check out her research. Spring, it was really a pleasure talking to you. I want to thank you for taking the time out of your schedule to do this. Thank you for everything you're doing there at your laboratory. And as we say on our show, thank you for helping to create the better tomorrow through your work. Very, very impressive.
It's been a pleasure. Thanks for helping us spread the message.